From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on June 26th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 100th episode of Matters Microbial. As you can see from this 3D printed logo of the podcast title by Dr. Jennifer Quinn, I am very excited by this milestone. I never guessed it could happen, and I am grateful to my guests, every listener and viewer, and Dr. Vincent Racaniello, the Podfather, for making it possible. Thank you all for spreading the good microbial word. I hope to keep going so long as there is interest from listeners and viewers. I recently traveled to Los Angeles for ASM Microbe, and I was well prepared to represent microbial centrism, as you can see here from my airliner seat. I posed with the ASM sign at the LA Convention Center, as seen here. While I was there at ASM Microbe, I gave a last-minute talk about demonstrations sure to excite microbiology students, seen here. And I also did a live podcast with Dr. Diane Newman of Caltech, seen here. The best part for me was running into some former undergraduate students, called Doc Martians, I didn't come up with it, who are now faculty members, seen here. We had the best time. Dr. Paula Wielander is a professor of earth sciences at Stanford University, and Dr. Krista Edmonston is an associate professor at the medical school at California Health Sciences University. I am so very proud of my undergraduate research students. I may not be the best researcher or a great educator, but I am a skilled talent scout, and I am proud of every single Doc Martian. I've sent about 29 or 30 to PhD programs. We've had a couple of podcast sessions discussing the idea of using bacterial viruses, bacteriophages, to combat bacteria that cause disease. There are many excellent reasons to prefer this approach over the use of antibiotics, which we have also discussed on this podcast. But what about non-human-centered phage therapy? For this reason, I'm extremely happy to introduce Dr. Heather Hendrickson, Associate Professor of Biological Sciences at Canterbury University in New Zealand, to discuss the work of her research group studying how bacteriophages can protect honeybees against bacterial disease. Welcome to the Quality Quorum, Heather.
Thank you, Mark. It's great to be here.
First, how long have we actually known each other? Did I really meet you while you were an undergraduate?
Did you meet me while I was an undergraduate? Where would that have been?
John Roth's lab.
Oh, yeah. I was definitely in John Roth's lab. So if you popped by at the University of Utah, I would have been one of the happy little workers in the laboratory.
Yeah, well, I met a lot of them, so I think that's the case, and that should give us all pause.
That's amazing.
And it was really nice to meet you at ASM Microbe and sit and chat. And here we are, actually outside the Wu-Tang Clan concert. I also have to say how much I love New Zealand, but you know that already.
I love New Zealand too.
I imagine so. So I have a quick question for listeners and viewers. You have a lot of things that you do in the research lab independent of this honeybee Panabacillus bacteriophage association. Would you give a thumbnail sketch of the other things that you study in the lab?
Yeah, so our laboratory has kind of two sides to it generally. And that has been historically a microbial evolution interest. And then interest in more applied projects, and that's really where the phages or the viruses of bacteria come in. So in our more fundamental science work, I guess the most recent thing that we've published was an eLife paper on the evolution of cell shape. So we managed to take a Pseudomonas fluorescens SBW25, which is a classic model for microbial evolution that Paul Rainey and Mike Travisano really developed in the 90s. And someone, not me, but Monica Girth, also in New Zealand, had managed to delete the MREB gene, which is considered to be completely essential in many microorganisms, or at least the bacteria really do poorly and have to grow in special media if they don't have this gene. And it's the ancient actin homolog, if you want to refer to it that way. And we managed to take that, which made huge spherical Pseudomonas cells, and then evolve it to be tiny spherical cells that had fitness that was equal to the original ancestral strain. So basically in 2 evolutionary steps, 2 different mutations, one imposed and then one allowed to develop through natural selection, we evolved spherical cells from a rod-like organism, which is pretty amazing. So that paper came out earlier this year.
You know, for listeners and viewers, this is a really, really big deal in general because bacteria have a cytoskeleton. And in fact, they're related to some of the cytoskeletal elements that eukaryotic cells, of which we are made, have as well. And many times I've run into people who aren't microbiologists who don't know that. But now every listener and viewer does.
Yep. It's pretty cool stuff.
I think that it's wonderful to have the 2 different sides of things. And I have a long-running interest in the maintenance of bacterial cell shape for all kinds of reasons. And so I'll put some links in the show notes to that kind of situation. And I think it'll be of interest to a lot of people. So Heather, go ahead, please.
I guess I'll just say, I think that probably, coming back to that idea of having worked in John Roth's lab as an undergraduate, that tying together molecular mechanisms and microbial evolution was really kind of a focus in that laboratory. And that's really where I got my start. So I think that has led to that, and also funding. So the fundamental work can be more challenging to fund. And so about 8 years ago, we started doing this more applied work. And I started talking about the idea of trying to find a way of using these bacteriophages to solve a problem in New Zealand.
And that we've talked about this issue of fundamental research and applied research, and most importantly, explaining what we do to the public, who's generally speaking, whose tax money pays for things. So this is something that's super important, and I'm glad that there's a lot of attention being paid to that. By the way, on Heather's website, listeners and viewers, there's a wonderful section all about public outreach. I will certainly link to that, and I urge people to read them, because this is something every scientist needs to do, and I applaud you virtually for doing so.
Thanks, Mark.
You're quite welcome. And John Roth is just you want to talk about a microbial hero. There he is.
Yeah.
So I think it's time to talk honeybees.
Let's do it.
So the first thing is, most listeners and viewers are aware that there are some issues with honeybees. They may have seen this in the media, but what you focused on is a particular disease of honeybees?
That's right. So you've mentioned Pennybacillus larvae. That's the organism that causes the disease that we're interested in. And the disease itself is called American foulbrood. And that name stems from where this was first found. It was first found in the United States, but it's now spread globally. And it is a huge problem in basically every place that has honeybees. In New Zealand, we've taken quite a strong approach to trying to eradicate this disease. And what that means is that if a beekeeper is finding any sign of this particular pathogen in their hive, then they have to board up the hive completely. So maybe first I'll show you an image, just what is American foulbrood. And so what you can see here is that American foulbrood is this pathogen. They form very long filamentous cells. So here we can see Penibacillus larvae on the right. And then what I didn't know before I started getting into this research, I didn't know anything about, honeybee development. And so I like to show a picture of what a healthy honeybee looks like in its development. And this takes like around 21 days, going from a tiny little egg that's laid by the queen bee. And that's it in its little kind of classic honeybee cell in a honeybee hive. And then it goes from that to what Irene Newton recently in her talk at ASM referred to as like the croissant. So they develop as these little almost croissant shaped larvae that live at the bottom of that cell, and they're fed by nurse bees every day, multiple times. So the nurse bees come and uncap the cell and then feed the little larvae and then close the cell again. And so if everything goes well, that little honeybee larvae will grow into a bee and get fuzzy and then come out of that cell. But if things don't go well, if something like a pathogen, for example, is put in along with the food, and particularly this American foulbrood causing pathogen Penibacillus larvae, then the honeybee larvae is melted from the inside out. And that is horrible for beekeepers, both because they really care about their bees and they have kind of an emotional, often attachment to their bees. But if in New Zealand you see any sign of this pathogen, and that's this central picture where you get this, you can put a stick into the center of the cell that's uncapped and pull out this smelly, horrible, ropey mass, which is the now full of the spores of Penibacillus larvae. So that is the true sign of American foulbrood.
So really quickly for listeners and viewers, and I run into this even with my students who aren't as clear as they could be about what a spore when it comes to bacteria is.
Yeah. So I don't have a picture of the spore on the slide here, but the long cells are the vegetative form. So those are the actively dividing. And as far as we can tell in nature, the only place that Penibacillus larvae goes into that vegetative state is inside of a honeybee larvae once it's been fed. And exactly right. So the spores are what are developed once that infection has completed. There are, I can't remember the number, but it's like on the order of billions of spores produced by a single honeybee larvae. And these are these tough, very small little dormant versions of the bacterium. Those dormant versions are then just highly transmissible. So this is a massive problem for beekeepers using their tools. They can then transfer those spores without knowing it to as many as 200 hives, right? So that is a massive problem. And then in New Zealand or another place like that, if you find this, every single one of those hives needs to be boarded up at night. So all the bees come back, you board it up, you put petrol in it, and you have to burn it. And that's the law in New Zealand. And increasingly, that's the way that people are approaching this problem in other places, because either beekeeper tools or hive tools or the bees themselves can spread those spores around. And those spores last at least 40 years in the soil.
Yeah. A lot of people are used to this idea with the human, well, not just human, but the disease-causing organism Bacillus anthracis, anthrax. Same principle with these spores, super tough. Nothing really works terribly well on them because they're, if not completely quiescent, they're almost quiescent in terms of metabolism, so standard techniques don't work very well. Things that make spores are a problem. And this, I won't say scorched earth, scorched hive approach is people can say it isn't a good thing, but I don't know what choices people have, because as you say, the spores are going to like easily spread to other places.
Yeah, that's exactly right.
Heather, I think many people, when they hear about beekeeping, heard about something in the news for quite some time called colony collapse syndrome. Is the foulbrood situation related to that?
So my understanding is that colony collapse syndrome or disorder was happening in a lot of places, but never actually took place in New Zealand. So we never faced that particular issue. But it was a very complicated set of pathologies that came about, from what I've heard, because of the use of kind of herbicides and those types of agrochemicals. So unintended consequences of the way that we were doing the kinds of all sorts of farming at the time. So we didn't have that issue, but when you start damaging the health of a hive because of those types of exposures, there will be fungi and viruses and bacterial pathogens that will all then contribute to basically the lost bees. So the bees that kind of wander off and then never manage to return to the hive. And that was a lot of what we were seeing during colony collapse disorder in the United States.
You know, it's interesting to me. I forget the fact. I mean, I know it, but in this context, it might be important. All those worker bees in a hive are clones. They're haploid clones. And that's so unlike what we're used to, right?
Yeah.
So it may be that particular genotypes are particularly sensitive to things. But as you say, given the amount of use of herbicides or pesticides or whatever side you like, I would not be surprised about that anti-emergent property.
Yep. So when I first kind of learned about this and started looking into the literature, there was some beautiful work that had been done by Yost and Penny Amy in the United States that showed that if you had bacteriophages against this pathogen, you could put the bacteriophages on a hive and they would actively protect that hive from getting an infection, even if it was in the same area. So they did a beautiful natural experiment showing that that worked and that prophylactic or kind of protective measure that you take in advance could work for as long as 4 months.
So we're talking about bacteriophages, as I'm kind of trying to show here. And these are viruses that attack bacteria. And they're very interesting. It's funny, though. Have people tried to use antibiotics to combat foulbrood?
In some places, that's legal. So in the United States, that has historically been legal. But of course, every time you use antibiotics, you're increasing the chances of resistance. And so now we have lots of the pathogen, Penibacillus larvae, that's resistant to the antibiotics that have historically been used. In New Zealand, antibiotics have not been allowed. If you find it, you must burn, which is why we thought we would take this approach in New Zealand.
I heard at ASM, by the way, that if you look at, I'm not going to say hive contents, but you look at certain types of commercial honeybees and you do a microbiome analysis, you find plenty of examples of antibiotic resistance genes. And this is because, believe it or not, antibiotics are used very, very commonly outside the human body. Agriculture is the number one user of it. So you know that when you have a situation where you use antibiotics, you can have antibiotic-resistant bacteria arising and sharing their genes. What's to prevent the issue with bacteriophage resistance?
So when we, and hopefully I'll come to this, but when we use bacteriophages wisely, then what we're going to do is we're going to come up with a cocktail. And what that cocktail does is it makes sure that, hopefully if you're doing it well, that you have multiple bacteriophages that are able to infect the same bacterial pathogen, but with different mechanisms. So different ways of getting their DNA in. And if you do that well, then the bacteria are not going to find a way of becoming resistant to both of those mechanisms in the same step. So you're going to have a much better ability to clear those infections or prevent those infections by doing really rational cocktail design.
You may or may not agree with me on this, and it's perfectly okay to disagree with me. Lots of people do.
Okay.
But one of the things that I tell folks is antibiotics themselves don't change because they've been synthesized and they're sprayed or given to patients, whatever it is you're doing with them. But phage can evolve And so it's kind of like they can change their side of it as well. And something to remind people, because this subject comes up a lot, is bacteriophages have nothing to do with eukaryotic cells. So that's important for people to remember, and they're literally everywhere.
So maybe I could show an animation that I made a few years ago to kind of describe what it's like as a phage in the world. So here we can see there's a little phage floating around, and you can imagine that this is like mucus system or a gut or whatever you like. But the bacteriophage is just this completely inert object that will only infect the specific cells that have the receptors that it requires in order to recognize its host. Then it injects its DNA, and that DNA is going to take over the machinery of that bacterial cell. And in as little as about 20 minutes in some cases, that bacteriophage can make thousands of copies of itself, if you're lucky, and then lyse that cell. And each one of those individual particles can then go on and fight that infection further or guard against that infection showing up further. That's in the case that we most want to find, which is phages that infect and lyse the cell or blow the cell up. In some cases, the bacteriophages will integrate into the DNA and hang tight, waiting for the right moment to go through that process of lysing the cell. And that's called lysogeny. So we have the lytic cycle, which is where they just blow them up, and then lysogeny, where they hang out in the genome sometimes, or sometimes as a little kind of plasmid-like bit of DNA in the cytoplasm.
You make me so happy, because I did most of my PhD work on bacteriophages of Cynorhizobium meliloti. So it's a big deal.
Yay. So I guess when we first started deciding that we'd like to try this project, one of the issues is that to find a pathogen like this, you have to go to where the pathogen is. And I am not a beekeeper in New Zealand. So we made this infographic that I wanted to show you. So this infographic was kind of designed by the local university's design shop in order to convince beekeepers that we needed their help in order to do this. And the beekeepers in New Zealand really came together for us. So we went and spoke to them. We went to the apiculture conferences, and we ended up with, you can see our phage sampling map. So here's a map of New Zealand, and you can see we had over 400 different samples come back to us from individual beekeepers who were happy to help by providing either soil or bee debris. And that allowed us to find a set of about 26 bacteriophages that we were able to discover eventually. But I have to tell you, like, I had been involved in phage hunts in the past, and, you know, sometimes you see a typical bacteria plate that has just so many plaques on it. So this is an agar plate that I think almost looks like the moon. What do you think, Mark?
Yes, it does.
Doesn't it? So this is actually a plate of Mycobacterium smegmatis. And this is a soil sample, I think, from compost that a student just found. In that single sample, so many different types of phages. And the reason that I say that is that this is a plate that should be completely covered with a thin layer of bacteria suspended in the agar. And instead, what we see is all of these pits, almost like craters in the moon. And each one of those, and you can see there's like many different types. There's some big spreading ones, and there's our little tiny ones. Those are each individual places where a single phage has infected a single bacterium and then gone through that process of lysing and lysing and lysing until it killed a whole bunch of different bacteria. But when we did this phage hunt in New Zealand, this was not what happened at all. So if we were very lucky, we would find a single plate in those samples that had tiny pinprick plaques. So they were very hard to detect, so much so that I can't even really show you a photo of what they looked like because they were so tiny. They just don't come out very well in photographs unless you really zoom in. So just imagine, instead of that like diverse, huge plaques that you find in some phage hunts, we just found tiny little pinpricks. And it wasn't because these were poorly diffusing phages. I can show you this picture of phage lilo. And this is an example of what our phages look like in this project. So this is Penibacillus larvae phage lilo. It was named by one of our many beekeepers. And phages like lilo, we finally managed to, after doing some adaptive laboratory evolution, we finally got good pictures and good DNA from these phages, but that took about a year. But they look a little bit like corn dogs if you're an American. There is a mycobacterium phage that looks like this that's called corn dog from the Hatfill laboratory. But they have this kind of elongated, I think what's called prolate phage capsid and flexible tails. So we would call them Cyphoviridae if we were getting into the nomenclature of phages.
I have to tell you that when you go on and I used to do phage hunts all the time, but for soil bacteria, specifically the rhizobia, and once I got one that had concentric rings in the clear area, and they were geometrical, not ragged. I've never seen anything like it. And after I left the lab during one of the moves, it got like tossed. So I have no idea what's behind it, which is too bad because it was beautiful.
There's so many interesting.
And there's so much media dependence. One thing that I want people to really remember about phage plaques in general is what limits them is that they're not effective against cells that are no longer growing, which is why the plaque does not continue to increase in size. So this is what makes them kind of exciting to look at. So you have some corn dog phage.
Yeah. And essentially all of the phages that we've found for this hunt have this same kind of look to them, or this morphology. Eventually, we managed to find a set of phages, and so I can show you this somewhat outdated now, but our Penibacillus host range. So this is the list of phages on the left-hand side. So you can see Callan, Dash, and Lilo are our top three, and then we go on to our others. We have names like Jacinda, so we have a phage named after Jacinda Ardern, the former Prime Minister of New Zealand, but also, Rosalind for Rosalind Franklin, and then we have a lot of names like Bob. I like the name Bob. And some of these are named by the beekeepers, of course. And then along the top of this table, you can see some of the diversity of the strains that we have. So we have representative strains from across New Zealand, and these come in 3 sequence types. But you can see that we still have a couple of strains from a particular area in New Zealand that we have never found phages for. And we're still very much interested in either evolving or discovering phages for those two.
Fascinating stuff. I think that listeners and viewers should know that there's a situation in New Zealand about importing and exporting biological materials. And the reason that this is important is that as you're saying this, I'm wondering how beekeepers are reacting to you having any Penibacillus larvae around.
Yeah. So before we were allowed to start this project, we had to reach out to the AFB pest management plan. So in New Zealand, when a pathogen is really considered a very high risk and something that we want to manage well, the government establishes through a mechanism that they have a pest management plan. And this is a committee of people who are stakeholders in the industry and who have broad experience in importation and that sort of thing. And they had to approve our application in order to even grow small volumes of this pathogen in our laboratory.
The other thing that listeners and viewers, and I'm not saying this is true for you or for me, but people might say, what is the utility of this? And so how much money does the bee industry bring in? And because that's threatened by all this.
Yeah, there are multiple ways of measuring that. One of the statistics that I found in New Zealand in particular is that there's an estimated $5 billion every year of our economy, which is directly affected by the health of our bees, either because of pollination or because of honey. And that pollination piece is a large part of that consideration in New Zealand, because we are a very strongly agrarian economy. We have a lot of sheep, as you may know. We also have a lot of cattle that are grazing on the fields, and the clover that's necessary to feed those cattle is all being pollinated by the imported honeybee.
So now you have to see how you're going to do the treatments. Is that correct?
Yeah. So once we have our collection of bacteriophages and we have our collection of strains, then we try to do this rational cocktail design. And so we came up with initially 4 different versions of cocktails, and we did something that we had seen in one of Graham Hadful's papers, which was to put the pathogens and also put the phage cocktail in 96 well plates at many dilutions. So I can show you kind of what the cocktail design strategy is, and this is just one example of one of our cocktail tests. So we have here in this image, we have our bacteria along the top axis at a decreasing concentration by 10-fold in each line. And what we've done here is we've done the experiment in these 96 well plates in kind of small volumes, and then we've taken a sample. And so what we're plating here is the survivors. So this is a phage cocktail that did really well. This looks like cocktail 1 on our 2017 strain. And so you can see when there's no phage, this is the growth that you would expect. That's that top line. And then we have decreasing concentrations of our bacteriophages, and you can see there are almost no bacterial survivors. So this is a lab-based test for how well the cocktails are doing at killing different types of bacteria.
You should have to have a high throughput technique to do this. And so absolutely. And I'll put links to some of the background to this, including Dr. Hadful's wonderful program that I know you've been involved with, and similar things.
Yep. So we did some cocktail testing, and we started to see really interesting things. We designed our cocktails without actually having the genomes for these phages. And we did that because we were in the middle of COVID-19 and we were still waiting for things to come back, and the student needed to move on with her project. So as we were waiting for the DNA to come back, we put together some cocktails and started doing some testing. And we saw, some really great results. But what we didn't realize at the time, and I think I have, so if we look at these phage maps, what we can see here is in 2 different rows, the genome maps for 3 different phages. So I have 2 of my Harrison cluster phages on top and 1 of my Vegas cluster phages on the bottom. So you can see this is representing the diversity of the phages that we've been finding. These 2 Harrison clusters are very similar, except for one little area. And then the Harrison cluster phages are quite different from the Vegas phages. And those Harrison cluster ones are like the ones that have really broad host range. And what was interesting in our, and I guess the other thing that I should say is this purple shading represents similarity. So those 2 different parts of those phages are very similar. If there's a white gap, they're not very similar. And what was really interesting to us is that our favorite phages that have the largest host range and can kill the most of our Pennybacillus larvae had sometimes this toxin. So that big purple box on the bottom row on the left-hand side is a big honeybee toxin. And so some of our best performing phages have a toxin that we're going to have to find ways of eliminating before we'll want to put these back out onto hives.
Bacteriophages can do this interesting thing where they can take hitchhikers from the chromosome to move things around. That process being called transduction, or more accurately, that we call that phage conversion, right?
Yeah. So they have picked this up at some point, and unfortunately, the 5 genes that are in the place that the toxin is in that top phage cause other problems when they're put into cocktails. So that little area seems to cause some kind of phage antagonism. So we have a couple of phages that look really amazing, and they're going to need a little bit more work before we can put them into cocktails in a hive.
I think this is amazing. And so you know that these are good prospects, and you've done, I guess you did some trial runs in the lab, and then you moved out to the fields?
We will be moving out to the fields. So we have these great, we have a wonderful collaborator in Ashley Mortensen, who is another American who loves bees, who lives in New Zealand. And Ashley is one of these people that are able to take larvae from a hive and put them into plastic dishes and then feed them and supply them with what they need and allow them to develop. So that is a great, safe space for us to test the effect of these phage cocktails. And we're going to be going back to do that again with a revised set of phages in October of this year.
Wow.
So we're very excited about that. And I guess one of the things that I should say is that we've actually managed to secure funding that allows us to do field testing. So with Pete Finneran of the University of Otago, also in New Zealand, we have managed to come up with what we've called the Adaptable Phage Solutions Program. And we've recently gotten about $9 million from the New Zealand government to progress phages for agriculture in order to eliminate the use of antibiotics or agrochemicals that might otherwise not be very sustainable in terms of our food systems. So if you can see here, we have kind of a plan for what we're going to be doing in the Adaptable Phage Solutions program. And that is we develop the phages, we go through discovery, rational cocktail design, and now we have a company that is learning how to scale up phages in New Zealand so that we can actually do this on our own soil, and that will also involve safety testing and then the money for field trials. And we are doing this. Pete is responsible for the kind of plant side. So he's involved in a couple of agricultural plants that are of importance in New Zealand. And my laboratory is doing the honeybees, obviously, but we've also got salmon aquaculture on our minds. And we'd like to use these same kinds of prophylactic approaches to reduce antibiotic use when people feel that they need to use antibiotics. The aquaculture sector in New Zealand is very much trying not to use antibiotics, but that also comes with trade-offs sometimes for what do we do when animals are not well. And that's really important to us all. So that's been a really fun project that we're doing in New Zealand, and we're really excited about. That's a 5-year chunk of funding to do this larger phage project.
I'm also a little concerned about what safety tests are necessary for the work that you do. That is to say, how do you evaluate the safety of a given phage procedure?
Yeah, so that's where these plastic dish experiments come in, at least in the first instance. So we can measure the health of these honeybee larvae that are in the dish, and we can either give them the pathogen or not give them the pathogen, or give them the phage and then the pathogen, or give them an important experiment, just the phage, to see how well do they do in terms of their development. And that's our first, measure. The other thing, of course, that beekeepers are going to be concerned about is if there's a phage present in the hive, am I going to see decreases, for example, in the honey production or decreases in their ability to pollinate? And so those are the kinds of things that we'll need to monitor when we do our blind field tests later in the program.
I do want to say parenthetically that one of the dangerous things that I hear quite a bit in public spaces is this idea of proving something that's safe. I mentioned this before. And that's where, if you work in any kind of pharmaceutical company, they have this term GRAS, genuinely recognized as safe. And those are some of the things that we really have to balance, because doing anything, driving your car, taking an airplane. All these different things have relative risks. And I think the important thing is to know what those risks are and evaluate them one to the other. I don't think anything is perfectly safe.
Yep. And I guess the thing that I should mention is that bacteriophages are in that category of things that are generally recognized as safe. So, we need to be cautious and monitor for things like that honeybee toxin that I mentioned. We certainly want to look at bacteriophage genomes to make sure that they don't have antibiotic resistance genes or other things that we could predict would be disadvantageous to increase the number of those genes in a particular population. But they are generally recognized as safe in the United States, and they have a similar category that I can't remember the name of in the EU. So they are coming around to being recognized as things that are already around us all the time and are generally recognized as safe.
They were kind of here first. In the news, there's been a lot of thunder and lightning over this term gain of function. And when you discuss extending the host range of a particular bacteriophage, is that at all related to that, or is this a tiny subset?
So in my experience, and you can correct me if I'm wrong here, I think there's a larger phrase that's generally used when we're worried about gain of function mutations. And that is that it's a pathogen and that we're increasing its function or its host range or whatever. And of course, with bacteriophages, they're not pathogens. So we don't have to worry about that kind of phrase in this context because they don't qualify. But this is, we're selecting for mutations that make them better at doing what they're doing, but they're not pathogens in the first place, and they're not becoming pathogens.
I think that's a good way to define it. I appreciate that. It's the only reason that I bring it up is that these things kind of the terminology gets into people's heads, and I see horrific things in the media from time to time.
Totally.
Something you don't know about me, Heather, is I worked for a number of years in, I'm going to call it a biotech company in San Diego, and we made xanthan gum as a suspending agent from Xanthomonas campestris.
Yeah.
So we'd have 100,000 gallon or more fermenters. And they would put the exhaust pipes in the sewer, which I told them wasn't a good idea. Because for listeners and viewers, sewers are a great source of bacteriophages. And they would get phage infections where all of that stuff would turn to froth. Everything would have to be sterilized. I would have to, quick like a bunny, find phage-resistant mutants of their production strains, and so on and so forth. But you see, that might sound like a problem, but I promise you, using antibiotics in agriculture is far worse because those genes can be transferred to other organisms quite easily. And when I say other organisms, I mean, imagine a harmless soil bacterium is resistant to an antibiotic and then can give that ability to things that can potentially cause disease.
That's exactly right. So I have a question for you about xanthan gum.
Yeah.
Are there always bacterial cells left in the xanthan gum, or is there a way of getting bacteria out of xanthan gum?
So that's an interesting question because we were always told not to answer it when we were asked that question. I never detected DNA in there, and keep in mind that the polysaccharide is going to co-precipitate like DNA. Okay. So when I tried PCR on it, I never saw it, but they put it through a lot of steps, so nothing living will be in there. But that's great. Okay. We'll talk about Gelan gum another time because I was looking for phage for Pseudoultramonas elodea at one point. Amazing issues. But anytime that you have Snackwell cakes, I help develop that.
Okay, I think I probably avoid Snackwell cakes. We don't have them in New Zealand.
I think that's fine. New Zealand's a wonderful place. It's the only place I've ever been that beer is cheaper than soda.
I'm going to have to go check that out. Is that still true? Interesting.
So this is a wonderful idea, and as always with phage therapy, and incidentally, I'll put a couple of links in, there have been some attempts to use bacteriophages to ameliorate, sounds like too much of a word, but to work with certain crop diseases. I know there have been spotty attempts to do that, and I'll throw some links in, but how do you envision treating the honeybee hives when you get to that stage?
That's a good question. I think we're going to have to, I mean, that's part of the project, right? Is figuring out delivery. And, I mean, I imagine trying a couple of different things. One is that because we want every nurse bee to give a little bit of bacteriophage to every larvae, I think one way to think about that is a sugar packet that you put in the top of the hive. And this is something that people use for delivering, RNA types of treatments if you're doing that kind of thing in your hives. So that's a thing to think about. But beekeepers really want methods that are compatible with the things that they're already doing at the hive. They spend about 80% of their time monitoring for AFB, but they don't want an intensive new process in order to prevent AFB. They want it to be like in line with what they're doing already. A lot of beekeepers are treating for like these little mites, these varroa mites that are like little vampires of honeybees. And so you don't, but those, they use often, at least in New Zealand, strips of chemicals. And that's not a great way of delivering bacteriophages. So either misting or a sugar packet. I sometimes imagine like a little water trough because honeybees always have to drink. Or what was the other? I can't think of any others right now, but that's definitely something that we need to think about.
But it's interesting because the bacteriophages will amplify themselves when they hit Panabacillus, the vegetative cells anyway. So it isn't necessarily true that you have to gut load these things up. It's not necessary. You just have to have them there. And for all we know, just dusting them with it is going to be good enough.
Yeah, exactly right. One beekeeper mentioned to us in a meeting that maybe if we had a phage dusting kind of on the outside of the hive, and it was charged in the right way. So bees, part of the way that they attract pollen is by charge interaction. So if we could dust and then have the right charge, then it would just stick to them as they go into the hive. And so there are lots of interesting ideas and definitely things to try to make this as easy. And it's also important that it's inexpensive for the beekeepers, or they won't engage with it. Although losing your hive because you found American foulbrood in it is also a complete loss of a hive. So those are the kinds of trade-offs that we'll be balancing as we go forward.
In a way, I'm kind of glad that there's not an attempt to sterilize the hives, because I don't know that you could really do that against that kind of spore load. Yeah. So we can make all the jokes about the X on the door during the Black Plague in Europe and all the rest, but I don't know what else the apiarists could do other than just destroy those hives. Yeah.
There have been attempts in some places to do things like to UV sterilize hives, but like you, I'm just not convinced. And I think, understanding people's eradication strategies and their mindset, and then working with them in the things that they're already trying to accomplish is going to be the way to make progress in terms of like an applied use of bacteriophages for this kind of problem.
What I like about this in particular, and it goes back to science communication, is it would be easy to say that folks involved with agriculture aren't necessarily interested in things that sound super advanced scientifically. Some are, don't get me wrong, but many are kind of old school, like my relatives. So with that in mind, what's really effective here is you're working with the shareholders. And we've seen this in several podcasts where people have wanted to work with the community, and this is no different. And being able to reach them and treat them as equal shareholders, and I hate that word shareholder, and I just used it, equal beneficiaries of the technology. And I think it's really wonderful.
Thanks, Mark.
And it's working.
Yeah, none of this would have been able to make any progress at all if it hadn't been for the beekeepers being willing to engage with us in the first place and send us these soil samples and these bee debris samples. So right now we have a very symbiotic kind of relationship with the beekeepers, which is appropriate for the subject.
I think that's a fantastic project. It must be very rewarding for you and your colleagues to be doing this.
Yeah, I guess I should say, so I have a wonderful lab group at this stage in New Zealand who's working with me. We are basically 100% doing phage work these days. So my adaptive laboratory evolution strategies for improving phages is the experimental evolution that I do these days. So here's a photograph of my amazing lab group in New Zealand. This is Danielle, and is the one furthest to the left. Danielle led this project from being an undergraduate, being a graduate student, all the way. She's now a postdoc on our research program. And then Rivers and Rewa and Breyer and Max to the right, and then Yan in the back row. And they are really, they are my happiest moment every week, is getting to talk to my my laboratory. So they are really an awesome group.
No, I've said it many times, at any level of research that you do, the people you're working with, when they have an interesting result or something unexpected, there's no substitute for that. It is so empowering to them, and it's so energizing to me.
Super fun.
So what's your So what do you envision in the future for your research plan?
So we are still in the middle of this big research program, but we're really encouraged by our kind of early results. We're getting more and more in depth with working with the aquaculture sector. I'm really interested in studying some of the fundamental things that I can see going on in this system. And that's actually, I guess I'll put in a plug for doing applied research. If you put in the time doing applied research and getting the funding that is, the slightly more low-hanging fruit, at least where I am, you can eventually get to a point where you start recognizing the more fundamental and fun, in a way, aspects of the project. So at this point, I'm really interested in studying this toxin further and understanding why it's in our bacteriophages. I'm really interested in why these phages make such tiny plaques in the beginning. Why do they seem so relatively unproductive? There's some really nice evolutionary questions, I think, in like kind of locked in with this system. And I'm really excited about exploring that further.
Many, many years ago, when I took the microbial diversity course at MBL, Marine Biological Labs in Woods Hole, Massachusetts, Abigail Salyers was one of my professors, and she's the one who told me, and this is many years ago, that when it comes to aquaculture, shrimp cultivation or prawns are a real problem, and they get sick because of the large populations, and then people would just dump antibiotics in. And
Yep.
she said she wishes there would be people working on bacteriophages as opposed to using antibiotics. And I've heard there's some folks that have done that.
Yeah.
But this, I think, is really going to be the future.
I will tell you, I was in a meeting, the Indian bacteriophage conference this last year. And there was, we were in Mumbai, and I was in, a cab at some moment with some of the other speakers. And there was somebody from the shrimp aquaculture industry who's been using bacteriophages in India for the last 30 years, which I thought was incredible. So there are definitely people taking those kinds of approaches. They don't always let consumers know that they're taking those approaches. And I guess with this project, I really like the idea of being more open about that, because, if you look at a little bacteriophage, these can look like an intimidating thing, even in this beautiful blue color. But I think the more, got a stuffy. Yeah. The more people know about bacteriophages and that they're already on us, in us, functioning in our lives every day, I think the less scary I hope that they will become. And that's part of the reason that we think that science communication is really important in our laboratory, because we want there to be kind of an open discussion about how these things work.
And honestly, most funding comes from governmental sources, which means they come from taxpayers. we have an obligation to explain what it is we do. The whole business of it's too complicated makes me very tired. And it makes me tired because it is possible to explain it. And the question is, do you want to spend the time to do it? And in exchange for support, I say yes.
Excellent. And here you are explaining everything to your micronauts.
Well, I hope so. I hope so. At least opening up some discussion about it. Anytime that you talk about phage therapy of any form, the first question you get is, can't it hurt people? And you have to deal with the fact that bacteriophages aren't recognizing anything about human cells. Then we get to the second level, which is, can you prove it's 100% safe? Now, I usually hear this from people smoking cigarettes, and I have a little trouble with that, but I put that aside. But I talk about it's difficult to prove a negative, right? The question is, have you done due diligence to show that something is, for the most part, relatively safe? And that's what the goal is. And it's so funny because in one context, people are very, very judgmental about it and very accepting in others. And the only solution to do exactly what you and your group has done is be completely open and straightforward about things.
That's what we're trying to do. There's, I guess, one other interesting aspect. So part of this program, which didn't make it onto my program diagram, is that we're doing a lot of stakeholder engagement. So we've recently made a really cute video, and we've done that with an organization in New Zealand that specializes in Maori engagement. So we're in a country where we have an indigenous population, and we are operating underneath the Treaty of Waitangi, which requires that we are respectful to that population and that they have recognized rights as the guardians of the country. And so we're reaching out to them specifically over the next 2 years with surveys and with this educational video to find out exactly how the indigenous population feels about where phages should be from, what phages can be used for, and what the future of this kind of prophylactic or even therapeutic process is in a country like New Zealand. So I think that's a really exciting part of being a scientist in New Zealand. I'm originally from California, and it's been an amazing place to learn more about good ways of interacting with an indigenous population. So I'll send you the link to that video so you can see the video by the TTW organization. It's really cute, and the accent is perfect.
My only thing when I was in Rotorua and went to some of the geothermal stuff, and they kept referring to things as being due to algae, and I shook my head, sadly.
Oh, okay. Well, you have to come back and see if they're still doing that.
Yeah, well, it happens, but no, it's a wonderful place, and I really appreciate what you're doing. I also wanted to note that you're wearing your 100 episode pin.
Indeed.
Which is wonderful. So do you have a little self-promotion here and there, Heather? You got to forgive me.
I understand.
So can you give everyone listening and viewing 3 things to remember from this particular session?
Sure.
Or 2 or 1?
Yeah. I guess I would say that bacteriophages are the viruses of bacteria, and so that's 1. Antibiotics and chemicals are, like agrochemicals, are not the most sustainable approaches to feeding the world and making sure that we have food for the future. And I guess 3rd, I would say we are attempting to come up with a protective phage cocktail that will protect honeybees from a devastating pathogen that they get globally.
You know what else I like to tell people about all this? And so I call microbes the small masters, first evolved, last extinct. And what I say, they have a lot to teach us because they were here first. And here's a great example of using bacteriophages to control populations.
We'll do our best.
Yeah. Now, really a couple of minutes on your route that made you into a microbial scientist as we end?
Yep. So that's a fun question because I get to see John Roth tomorrow. So I am going to be dropping by my OG mentor's house to see him and his wife. I was an undergraduate at the University of Utah, and I was actually a married Mormon choir director and a biology major. And I kind of by accident stumbled into John Roth's laboratory for microbial genetics. And I didn't know that he worked on microbial evolution. And we got into a conversation, and I realized I had been reading Scientific American articles about the very thing he was working on. So I guess I hadn't really done my research before I got this job. But John kind of dragged me from the secretary's desk into the laboratory and just had a huge effect on the trajectory of my whole life. So today I am a happily divorced scientist living in New Zealand, and none of the amazing things that I have been able to experience and the great experiences I've had in science would have happened if I hadn't accidentally ended up working with John and having such an incredible mentor. And I'll always be really grateful.
So there's a saying, that there are no coincidences. And if you're of a religious persuasion, they say that God whispers. We can say the universe whispers. We just have to listen. And knowing John Roth, who I'm quite sure doesn't remember me anymore, I've only had a few interactions with him, but do tell him hello.
I will.
In case he does. One of the term thought leader isn't enough for John Roth. And I'll put some links to stories about John Roth in the show notes as well.
Just an incredible scientific leader and such an interesting character. I think he really made it clear to me early on that we don't all have to be the same in science. He is really a remarkable character and has just, led to so many scientists. And I think we are all quite different. And just couldn't thank him enough.
Well, it's been wonderful chatting with you, Heather. And it feels like we just talked because we did in Los Angeles, right?
Yeah.
And I want to thank you for your time then and today.
So good to see you, Mark. And I hope that we can get you down to New Zealand very soon. I'll work on it.
I hope so. I think it'll be a lot of fun. Thank you so much, and my best to you and your research group and family.
Thanks, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Heather Hendrickson can be found in the School of Biological Sciences at Canterbury University in Christchurch, New Zealand. Many thanks, as always, to David Renata for superb editing and Reaper Clark for the wonderfully quirky music. Again, thank you to every guest, listener, and viewer for helping me find my microbial voice. I am so very grateful on this 100th episode. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.